Hello, everyone. Good afternoon, and welcome to today's edition of Enhanced 3D Genomics, the 3D Regulatory Genome. It's a great pleasure to introduce Argyris Papantonis today. Welcome him as our speaker for today. I've known Argyris since, I don't know, actually probably 2010 or something like this, since our early postdoc days. We both essentially haven't changed since then, but we were both working on transcription factories back then in Oxford and Cambridge, respectively. Argyris studied biology at the National University of Athens, where he also did his PhD in the lab of Professor Rena Lekanidou. And in 2008, he moved to Oxford for his postdoctoral studies with Professor Peter Cook, who actually discovered transcription factories at the Sir William Dunn School of Pathology. And after 5-6 years there in 2013, he moved to Germany, initially as a junior group leader for chromatin systems biology of the Center for Molecular Medicine in Cologne. And after another 5 years in 2018, he was recruited to the medical faculty, the Georg August University of Göttingen, where he is currently a professor for translational epigenetics and genome architecture. And welcome, Argyris. Thanks for.
No, thank you for inviting me.
For being here. I very much look forward to your talk.
So then I should do my bit and try and share my screen.
Yeah, let's try.
Let's do that. All right. So there we are. I think you can all see that. Right?
Yeah.
And okay, so off I go then. So, all right, officially, thanks again, Stefan. It's also very nice to see you after a while. It's been a while. And I've slightly changed my sort of more sciencey title that you saw on the slide with a rather simple questions. And that question would be, why would senescent cells decide to cluster CTCF molecules that should otherwise be, throughout their nucleus and chromatin? And this is the question that we'll try to address today. So senescence is a very interesting physiological process, and it's actually intrinsic to the vast majority of cells in our body. So there's a sort of bright side to it, which is usually referred to as acute senescence, which is the triggering of an anti-proliferating program that comes as a response of DNA damage, hyperactivation of oncogenes, or simply the exhaustion of the replication potential of cells. And what this does is it sort of keeps at bay growth stimuli and controls the growth of individual tissues and organs. And also early in development, this has a very interesting role in how different parts of the body, like our fingers, would develop, or in wound healing and acute injury. However, with time, as tissues and organs age, you have an accumulation of these senescent cells that create niches that are very rich in inflammatory stimuli because of the secretome of these senescent cells. And this also makes clearing from the immune system worse. And it's actually now very well understood that the presence and accumulation of senescent cells is one of the hallmarks of the onset of actual organismal aging. So we wanted to understand how would a cell commit to this fate of senescence. And of course, if you just simply look at a dish where your cells will senesce as you keep growing them, you very quickly realize that this comes about pretty stochastically. But of course, and this looks like generally a stochastic process, as is aging for the most part considered to be. But if you were to consider an individual cell, we hypothesized many years ago that there could be early and potentially deterministic events that commit the genome of individual cells to this fate of senescence. And if this hypothesis were true, then these events would be driven at the level of chromatin, either chromatin regulation or organization, and they would be driven likely by chromatin binding factors, which would make them detectable, first of all, so decipherable, and perhaps reversible or druggable in an effort to slow down the natural process of cellular aging. So what we discovered in a paper that's already now been 7 years, published 7 years ago, is that you could easily tell apart a senescent cell in a population of cells just by looking down the microscope and asking whether this protein, HMGB2, is present in its nucleus. So generally larger cells that do not have HMGB2 in their nuclei, and you can see 3 different examples of 3 very different cell types here: mesenchymal stromal cell, a fetal lung fibroblast, and an endothelial cell. They all sort of go into the senescent state by losing HMGB2. And at the time, we thought that this would be a very interesting protein to look at, because these high mobility group box proteins, first of all, they've been known for many, many decades because, and the name sort of reveals that, they were initially identified in nuclear extracts many, many years ago, just because they run faster than most other proteins. And also because they were very, very abundant, so they gave a very big blob as a band on a coma-sustaining gel. They are very broadly expressed in different tissues and organs in our body. HMGB1, HMGB2, these are the 2 major proteins. They're also pretty much alike one another. And it's very interesting because they're very conserved, and they are the most abundant nuclear proteins after histones, with HMGB1 calculated to be 1 for every 10 nucleosomes in the genome. So we're talking about many, many hundreds of thousands of copies. And then from this huge abundance that you have in a normal proliferating cell, you go to essentially 0 when the cell senesces. So we focused on HMGB2 back then, and we performed a very simple knockdown. And we discovered that just by taking out this one, albeit very abundant protein from the nucleus, you had 3 very profound effects. The first one was that the knockdown reduced the overall transcriptional output of the nucleus by 15 to 30% roughly. And this is by staining nascent RNA. The other is that it induced almost immediately, so within the 48 hours of this knockdown, a dramatic shift in heterochromatin. So we had a loss of the facultative heterochromatic mark of K27 trimethylation, a significant drop, and this seemed to be replaced by an increase in constitutive heterochromatin marked by HPO1 alpha. But what was very surprising to us, that no one had ever seen before at that point, is that when we knock down HMGB2, here you can see a cell with a knockdown, and here you see 2 cells of control. Here's HMGB2, it's essentially everywhere in the nucleus. Here it's knocked down. CTCF, which is a major protein for 3D genome organization, a major player in this, actually goes from this kind of appearance to this very dotty appearance in the nucleoplasm. And this is exactly what you see when you look down the microscope at a senescent cell that is also larger, because also there's a lamina deterioration, and you have a flattened nucleus and so on and so forth. But CTCF has this very distinct clustering that appears. And this is not aggregates, which tend to happen as cells age, because you can actually wash out all the non-chromatin bound parts of the nucleoplasm with a simple CSK extraction, and still these clusters will stay behind. So at the time in 2018, and here's a super resolution image of what this phenomenon looks like, we defined HMGB2 as a protein, the presence of which actually dictated what the distribution of CTCF will be inside the nucleus. And we call these clusters senescence-induced CTCF clusters, or SICCs. And we argue that they contribute a genomic hallmark for senescent cells. And I think you can see here that B2 and CTCF are not overlapping that much. They seem to be sort of interdigitating in the signal. This is a very thin optical slice on a STED microscope. And you can see here these very large clusters that presumably contain many tens of CTCF molecules. But this work, of course, was descriptive of the phenomenon and of the trigger to the phenomenon, which was the loss of HMGB2, but it said very little about the molecular basis of SICC formation. So what is the molecular basis that allows CTCF to do this clustering, which we have not seen in other contexts? About the relationship of B2 and CTCF clusters, because B2 and CTCF are actually 2 proteins that do not occupy the same positions on chromatin, and they don't interact physically with one another. So how would the loss of one affect the other? And then, of course, is there a functional importance to the formation of these CTCF clusters, or are they simply a byproduct of the cell entering senescence and, okay, not dividing anymore and nearing the end of its lifespan? And these questions were actually tackled by a trio of very talented students. So Spiros, who has actually graduated, did all the experimental work that you're going to see. Yaji, who also recently graduated and got her PhD, did all the splicing analysis you're going to see. And Ilya, that is coming close to graduating, did all the other bioinformatic analysis that you're going to see. So a first small interlude here. The problem that you have when you study senescence in vitro, like we do, is that you have to wait until the cells reach this point of senescence, right? And of course, every cell, as I said, will reach that point at a different time, and this gives rise to heterogeneity and so on and so forth. So we wanted to remedy that, and we were lucky enough to find a molecule that is called ICM, or inflachromine, that was published in 2014, which, when repurposed, this molecule is supposed to directly target HMGB2. So when repurposed and thrown onto primary fetal lung fibroblasts from humans, which are a very, very well-known model for senescence, it will give you, within 6 days of treatment with 10 micromolars, a very homogeneously senescent population in a short, defined time frame, where you would have loss of HMGBs, the activation of P21, a very well-known marker of senescence, the shift in heterochromatin just like before, and of course this beautiful large CTCF clusters in the nucleoplasm. And then when we look at gene expression, this is one of the many things you'll find in our aging cell paper that describes the system in much detail and also offers lots of genomic resources for people that want to use it. We actually find that there's a very nice correlation to the gene expression profile of replicative senescence, which is what fetal lung fibroblasts will get to once they've been cultured enough, and not so much to other types of senescence like oncogene-induced senescence, because this is not the context here. So this is going to be our system from now on. We'll be treating the cells with the drug for 6 days or not. So what is the molecular mechanism? So we started with a rather simple experiment. We pull out CTCF from young and from ICM treated, so from senescent cells, and we ask, are there proteins that come down with CTCF significantly more in the context of senescence? The answer is yes. There are 12 of these, and they belong to different categories. There's proteins that seem to be transporters, there's diffuse proteins, there are splicing factors, there's P62, that is a transcription factor known to be linked to autophagy, and so on and so forth. Most of them are actually increased in expression with senescence. Only BANF1 seems to be reduced. So we wanted to look into whether these factors actually had anything to do with CTCF clustering. For some of them, we cannot do functional assays because they're part of large structures or the nuclear skeleton, and it's difficult to knock them down without having detrimental effect to cells. So we ended up studying these factors that you see here. So P62, BANF1, FMR1, SRM2, and ataxin-2. And to cut a long story short, 3 of these 5 proteins have no effect when we knock them down in these cells. So you have a senescent cell that has CTCF clusters, you knock out these proteins, nothing really happens to the CTCF clusters. However, 2 of them, BANF1 and SRM2, seem to have very interesting effects. I'm going to start with BANF1. So this is a very small protein. It's rather structured, so it hasn't got any of these intrinsically disordered domains that will become important in a bit. And there's a very nice paper from the Gerlich lab in Vienna, where they've shown that it actually acts as a molecular glue for chromosomes during mitosis. So the set of chromosomes that needs to go to each daughter cell is actually coated by BANF1 molecules that act as a molecular glue onto chromatin, bridging it to proteins of the nuclear envelope. And if you don't have BANF, you actually can't keep the set of chromosomes together, and you have mitotic catastrophe and lots of defects. So when we knock down BANF1 from senescent cells, you can see here, this is our knockdown, this is our control, we completely lose the ability of CTCF, well, almost completely lose the ability of CTCF to cluster. So BANF1 seems to be necessary for clustering of CTCF in the context of senescence. However, when we overexpress BANF, and you can see here two cells, this one, we've overexpressed a GFP tag BANF1, and in the neighbor cell, we don't. This is not sufficient to induce senescence. So BANF1 seems to be important for allowing CTCF to cluster, but it doesn't seem to suffice and cause clustering in the absence of presumably other factors. Now, the second candidate is SRM2, and this is a very, very different protein. So this is very large. It's almost 3,000 amino acids in length. It's highly unstructured, as most RNA binding proteins are. And it's a very well-known component of the nuclear speckles, which are these phase-separated entities, which are sort of hundreds of nanometers to micrometers large in the nucleoplasm. So what happens when we look at SRM2? Well, if you just simply stain for SRM2 or for speckles using this marker, SC35, you actually find that the CTCF clusters are on top of speckles, and you can see this here in this line plot as well. So every time you see a speckle in red, you actually see a CTCF cluster on it. There's no CTCF clusters outside of speckles. And to give you a sort of high resolution picture of that, this is a STED picture of what a young cell, so DMSO treated, would look like. You can see the different speckles here in red, and you can see in green the signal of CTCF very nicely, surrounding them every now and then, overlapping the speckles, but very little, right? This makes sense. So if we look now at a senescent cell, this changes dramatically. You actually have the speckle. You can see here the SC35 signal, and CTCF seems to overlap the speckle heavily, although to be a bit offset to the bulk of the signal of the SC35. And you can also see some dots here, probably representing smaller clusters of CTCF. We can now also manipulate the speckles. So there are drugs like tautomycin and cantharidin that allow you to either disperse speckles or fuse them together. And when we do that in senescent cells, we actually see that the CTCF signal very closely follows either the dispersal or the fusion of these speckles, which means that the two entities, if not mixed, they definitely crosstalk very tightly. And just as a sort of spoiler, I don't think that they are one entity. I actually think that the CTCF clusters form either on top of speckles or sort of semi-mixed with them. Now, if we knock down SRM2 like we did before for BANF1, we have a full abrogation of the ability of CTCF to cluster. We don't see any clustering whatsoever. So SRM2 is much like BANF1, necessary for the formation of CTCF clusters. And this is not because taking away SRM2 changes the levels of CTCF. You can see that the levels are the same. It just causes a very strong redistribution within the nucleus. Now, the question is it sufficient for SICC formation? So what we've done here is we thought, well, speckles are phase-separated entities, and SRM2 is a disorder protein. So maybe this intrinsically disordered domains, you can see one here, highly disordered, according to the to this UPRET test here, and highly disordered here. Maybe this is what plays a role here. And to be very honest with you, what we try to do is we try to clone the whole IDR. So the N-terminal, sorry, IDR here. This should say C-terminal actually. So the N-terminal IDR here that contains the RNA bind domain of the protein, and the C-terminal IDR here. But this was very difficult because these are highly repetitive regions. So we were able to clone just a 68 amino acid long part of the RNA binding domain and an equivalent part of this C-terminal IDR. When we overexpress this very short 68 amino acid long RNA binding domain, you can see that the overexpression, here's two cells. We overexpress in the left cell. The right-hand cell serves as a control when we add DOX. You can see that these blobs form, and also that we have lots of signal in the nucleolus. You can see the nucleolus here. And these blobs that are outside the nucleolus actually overlap speckles, and they give rise to CTCF clusters. And we see no clustering in the nucleolus whatsoever, despite the fact that, this small peptide also accumulates there. Is this specific to the RNA binding domain or not? If we use any IDR, we will have the same effect. We do not. So when we overexpress this C-terminal IDR, we actually, so in this cell here, but not in that one, that does not drive the clustering of CTCF at all. So it seems like a very specific part, which is the presumed RNA domain, RNA binding domain of SRM2, suffices to drive clustering in the context of a young cell where CTCF should not normally cluster. We can also do another experiment, and this is my favorite one actually. We can knock down SRM2 from these cells. And once we do that, when we do the knockdown in senescent cells, we now have no CTCF clusters whatsoever. And now in the absence of structured speckles, because we've taken away one of their major architectural components, which is SRM2, we overexpress just 68 amino acids of this 3,000 amino acid long protein. And this is enough to give rise to small but discernible speckles. Again, you can see them here. This 68 amino acid peptide actually goes to these speckles that now form, and CTCF readily now clusters on these speckles. And you can see the quantification here. So this means that this is not just sufficient to sort of reform the speckles because they collapse a bit in its absence, but also to give rise to CTCF clusters. Just 68 amino acids in the absence of any other SRM2 in these cells. So if this is the case, so if the clustering occurs on the surface of speckles, and if you only require this RNA binding domain, how would HMGB2 come into play? Why would the loss of HMGB2 actually be relevant to CTCF clustering on the surface of speckles? Well, we revisit some old data where we did a very simple co-IP mass spec of HMGB2 from young cells. And we found that the vast majority of known speckle-associated proteins, including SRM2, are direct interactors of HMGB2. So we started thinking that it could simply be a competition. So HMGB2, which is super abundant in the nucleus, likes to interact with SRM2. And in its absence, now CTCF can interact strongly with SRM2 and cluster on the surface of speckles. So we wanted to test whether we can actually rescue this. So we had in the past shown that if you just overexpress full-length HMGB2 in old cells, so these are ICM-treated cells here, senescent cells, you actually can disperse many of the CTCF clusters. Now the question is, do you require the full-length protein, or are some parts of the protein enough to do this rescue? And we looked at the classical HMG box DNA binding domain of HMGB2 and its unstructured acidic tail. If we overexpress just the HMG box, this is not enough to rescue the clustering phenotype. But this acidic tail, which is, again, just 72 amino acids in this case that we're overexpressing, suffices to interfere with the interaction between CTCF and the speckles and dissolve the CTCF clusters. So it seems that there is an interplay between different IDRs, different intrinsically disordered regions, either from HMGB2 or from SRM2, with the two IDRs that are in the N and the C terminus of CTCF that control this interplay and the clustering. As I said at the beginning, CTCF is very important for 3D genome organization, which is also the sort of core idea of this seminar and of the company that is being run now by Stefan. And you would imagine that if you physically cluster CTCF, which is a protein, by the way, that likes to spend lots of time on chromatin, you would rearrange the 3D genome. So we used microC to actually address this question. So here you see a microC map. I assume that many of you are going to be familiar with this, but I'm going to sort of take you through. So we're looking at a map, an interaction map from this small bit of the chromosome that is about 2 megabases. And you can see here two triangles that depict the same region, these 2 megabases, in control and senescence conditions. And this heat map, essentially, every dot on this heat map gives you the interaction value between the 2 pieces of the diagonal that it connects. So this dot here is a loop between this bit of the chromosome and that bit of the chromosome. And I've put highlights, I've put these circles here to show you that there are multiple loops that come about in the senescent cell. You can see these dots inside the green circles that are either very weak or not at all present in the young cell. And these represent CTCF loops. So in the context of senescence, you have the formation of multiple new CTCF loops in the cell population. And these, as these graphs, average plots here show us, tend to be stronger loops. There's many thousands of them, and they tend to be stronger. And they also tend to be, on average, longer than the loops that you see in a young proliferating cell. So the idea here is that in the state of senescence, presumably the clustering of CTCF allows the formation of many new loops that are on average longer in size. What is also interesting is that although the average occupancy of CTCF on chromatin drops a bit in senescence, the positions, the anchors, as we say, of these loops, so the positions that come together to give rise to these loops, are actually strong CTCF sites. And they bring about more insulation. So they insulate the contacts within the loop from contacts that come from left and right of these loop. So they do give a very different 3D genome organization to these cells. However, this is an experiment that is done on a population of cells, and it's difficult to say whether these loops all come about in the same cell or whether they come about in different cells, and we just see the average of them. So what we did is we paired up with the lab of Jan Ellenberg at EMBO, and especially with Kai Beckwith, who led all these experiments that I'm going to show you now, to try and look at 3D genome organization of single DNA fibers inside single cells. And to do this, we do something that's called DNA tracing. So essentially, what Kai and Jan are doing is they're probing very tightly with lots of probes. They trace chromatin inside single nuclei. And then by just consecutive imaging, they can tell us what kind of structures we can see. And what is interesting here, and this is why I put this up, is that we can have cells, like here's a young cell on the left, here's a senescent cell on the right, where we have stained for nuclear speckles in green. And you can see the tracing of chromosome 2 in magenta here. And this is a tracing that is very low resolution. So it has a 1 megabase resolution, so we wouldn't see loops. But you can see in light blue here and there, and also here and there, the 2 alleles of chromosome 5 that are being traced at 12 kb resolution. So a resolution that is very close to microsy. And this is what they look like. So if you take all these high resolution traces from this 1 megabase region on chromosome 5, this is the region that we're probing, you can see the average 3D genome organization from 450 cells here in control cells, in proliferating young cells, and 217 senescent cells. And I think it's evident just by looking at this that there's a profound reorganization with these longer range loops forming. And if I compare this to microsy data from exactly the same region, I think you can all appreciate that they look pretty much alike. You can see the long range loop here, which you can also see in the DNA tracing data, and so on and so forth. But this gives us the ability not to look at average of these 400 or 200 cells, but to actually go to single fibers from single alleles and then ask, what do they look like? And this is what they look like. So in young cells, you would have this more open conformation where a bit of the traced chromatin is actually in the vicinity of a nuclear speckle. You can see it here in green. There's another one, right? You can start from the dark blue and trace the whole thing all the way to the light green edge. But then when you look at these conformations of this 1 megabase region on chromosome 5 and how they change in senescence, you see these scrunched up multi-loop structures that actually are on the surface of speckles, almost invariably. And this allows us to do some measurements at the single cell and single allele level, which goes something like this. In senescent cells, we have more contacts forming, and you can justify this because it's a more closed interaction. We call a contact anything that is less than 200 nanometers away from, 100 nanometers away from one another. The loop sizes that form are actually longer, which is exactly what we saw also in our micro-C experiments. And most interestingly, you have the formation of multiple nested loops, as if you were forming rosettes with many loops on the surface of these speckles, which is intuitively what you would think happens when you bring lots of CTCF-bound to chromatin on the surface of speckles when these clusters form. And the last measurement that Kai could do for us is that we can look at the contour size, so the actual length of this region that we're looking at. And this also increases, which goes very much along with the idea that you have fewer nucleosomes in senescent cells, and these fewer nucleosomes actually allow for a more stretched out contour length of chromatin. So now we've established, I think, that we do see these multi-loop structures. They tend to be on the surface of speckles, and we can see it both in micro-C and in these sort of single allele tracing experiments. And the question one needs to ask now is there a reason to this? Is there a functional implication of these restructuring of the genome and clustering of CTCF for senescent cells? Or is it just a byproduct of these cells losing HMGB2 for some other reason? So back in 2021, we had combined nascent RNA sequencing with proteomics and RiboSeq in this transition from young cells to senescent cells to actually ask the question, at which level of gene expression regulation is most of the changes that we see at the protein level? Where do they manifest? And to cut a long story short from this analysis, the vast majority of changes in this particular model of senescence can actually be explained by changes at the level of transcription. But we had never looked at the level of splicing. We only looked at the expression levels of transcripts. And of course, nuclear speckles, or the proximity of active genes to nuclear speckles, has now been linked to differential choices in splicing. There's a very nice paper, for example, from a year ago from Mitch Goodman that shows that if you tether a gene close to the nuclear speckles, you have a very different splicing pattern than if you tether it away from them. So having this in mind, Yaji, who did all these analyses that I'm going to show you, started thinking about splicing changes. And for example, if you look at ICM-treated cells or senescent cells, they show many thousands of alternatively spliced genes that change by at least 20% in this case. This is the cutoff we had. And many of these splicing changes, you can recapitulate simply by knocking down HMGB2, which, as I showed you at the beginning, is sufficient to cluster CTCF onto speckles. And then what you can do is, and you see these plots again, so this is why I'm going to explain them now, you can correlate individual splicing events. So every dot here on this plot is not a gene that is differentially spliced, but it's an actual event. So this is, for example, alternative usage of 3' ends. So every dot represents a specific coordinate, a specific part of the 3' UTR that's alternatively used in these transcripts. And I think you can appreciate that there's a very strong positive correlation between the changes upon knockdown of HMGB2 and the changes imposed by bringing cells into senescence. What is also interesting is that if you look at these positions, especially the shared ones, they seem to be marked by, on chromatin, by signal of HMGB2 and or SON binding, which is a marker for proximity to speckles. So what we think is happening is that many of these alternative splicing choices occur at positions that are marked by HMGB2. And once it's lost, then the splicing choice at that position seems to be affected. And this doesn't only apply to chromatin, which is what I'm showing you now, but HMGB2 also has a true RNA binding capacity, and we have clip data that show a very similar picture to what you're seeing here. So the binding of B2 on chromatin or on RNA seems to correlate pretty well with the positions that become alternatively spliced. Okay, so what we know is that if you treat cells with ICM, you engage, or you commit to the fate of senescence, but you also form CTCF clusters. But then there are three types of interventions that you can do to senescent cells and preclude the formation of CTCF clusters. And you can knock down CTCF itself, you can knock down SRM2, or you can knock down BANF1. So we did this individual knockdowns, and we did RNA-seq and mapped the splicing changes that come with it. And what we saw is that there's a significant overlap between each of these knockdowns and their alternatively spliced mRNAs and those that come about in senescence. So we tried to decode that. One way to do this is to use a piece of software called isoform switch, which categorizes the extent to which your alternative splicing changes affect isoforms, known or de novo isoforms in your catalog of mRNAs. And you can see that when we add ICM to cells, we have about 1,000 transcripts that have differences in isoforms. And you can see that you have significant, for example, reduction in the usage of particular alternative donor sites, of intron retention, of core exon usage, and an increase in changes that involve either the 5' or the 3' end of transcripts. If you just knock down HMGB2 that also allows CTCF to cluster, you have a same overall profile. The same tendency to have less of AD and intron retention, more of these two. And if you look at the genes that are behind these events, you actually find that they are enriched for GO terms that are very, very relevant to entry into the state of senescence, like cell cycle control, chromatin reorganization, microtube organization, because you don't go through mitosis anymore, and so on and so forth. So these seem to be convergent changes to the alternatively spliced transcriptome that correlate with an entry into senescence. So what happens when you knock down proteins that don't allow CTCF to cluster anymore? Well, what we see is that for the most part, and you can see it most clearly in the CTCF knockdown, but also in the others, you have an undoing of the patterns that we see. So for example, you have less of these changes and less of those changes here. And again, in the case of CTCF, everything almost goes back to the levels that we had in the control cells. But again, this is an overview. It would be good to look at individual events. So let's do that. So as an example, we can look at exon skipping and exon inclusion events of core exons. And here you can see the individual alternative splicing events that happen when you knock down BANF1 or when you knock down SRM2. These two knockdowns, the common denominator is that they don't allow CTCF to cluster. And you can see here a very strong positive correlation of these events. And now, if I correlate the knockdown changes in alternative splicing to those imposed by ICM, I have an almost fully inversed anti-correlation. And this is the case for knocking down SRM2 or CTCF. This simply means that changes that are imposed by ICM are fully reversed when you knock down any of these three factors, again, the common denominator of which is the loss of the CTCF clusters. So is this, apart from changing the splicing program, is this significant for the physiology of these cells? Well, if you take a young cell, you can see it here, and you start knocking down, this is a control knockdown, you see that these cells will happily grow in culture. But if you knock down SRM2, CTCF, or BANF, they will stop growing. And for CTCF and BANF, this is known because the CTCF knockdown will cause S-phase defects. The BANF one, as I said at the very beginning, was already shown that it has mitotic defects, so you don't grow well. But the SRM2 as well will give you the same effect. What happens if you do the same knockdowns now in senescent cells, where they shouldn't grow because they're senescent, and you don't have cell division anymore, right? Inherently, they're senescent. So what happens is this is the senescent cells, and every single one of these perturbations that, again, do not allow CTCF to cluster, give you a delay in the onset of proliferative arrest in these cells. So we think that the easiest explanation to this data is that the integrity of these CTCF clusters is actually a functional event that is required for executing alternative splicing and committing these cells fully to the fate of replicative senescence. So this is all nice and good. We feel that we have deciphered this event, so we mechanistically understand how it comes about. We understand now its relationship to 3D genome organization and also to alternative splicing choices and how these are directly related to the commitment to senescence. But of course, this creates a number of questions that need to be addressed in the near future. First of all, is there a role for RNA in SICC formation? Speckles are very rich in RNA, but at the same time, senescent cells produce less RNA overall. So how does that work there? Are there isoforms or post-translational modifications of CTCF that contribute to the phenotype that we see, to the clustering? Are these senescence-induced CTCF clusters present in any other context? And of course, could all this become relevant for clusters, for cancer, sorry? So could we learn something from the senescent model that we can sort of induce in the context of cancer to constrain cell proliferation? So with this, I'm going to stop, and I'm going to thank the people that did the work. Spiros, again, as I said, the very first one, but also people that are now following this up in the lab, like Sia and Tassos. And of course, the people that have helped out a lot with this work. Christophos, that helped with analysis. Marike has done chipmentation for us. Jan and Kai, who did all the very complex DNA tracing. Carsten for all his advice on phase separation, and Leo for the clips. And with that, I'm happy to take questions.